So this video will discuss the cell cycle and mitosis, and then the next video will go into more detail about the control of the cell cycle. So the point of the cell cycle is to make more cells, not surprisingly. And the cell cycle is sort of like a life cycle of the cell. It begins when the cell is produced, along with usually a sister cell, and it ends, the end of the cycle is when that cell then divides itself and produces two new cells. So before we can talk too much about the cell cycle, we need to know a little bit about chromosomes and chromosome structure. And we're going to focus here on eukaryotes. This is what's known as a karyotype of humans. It shows all the chromosomes from an individual human. And you can see humans have 22 pairs of autosomes, meaning chromosomes that don't determine the sex of the individual, and one pair of sex chromosomes. And you'll notice that the autosomes, the members of the pair are extremely similar to each other, and those sex chromosomes are actually quite different, and that will become important when we talk about meiosis in the future. So eukaryotic chromosomes are linear. You'll notice that there are generally 2 copies of them. That's pretty common in eukaryotic animals. And that makes us a diploid, meaning we have 2 copies, as opposed to a haploid organism that only has 1, or a polyploid organism that might have multiple copies of each chromosome. So this is a bit more close-up picture of a chromosome, and you'll notice, interestingly, that it looks like it's 2 chromosomes. It's actually 2 DNA strands stuck together. And that's because the easiest time to visualize a chromosome under the microscope is actually at this point when it's already duplicated and the cell is actually about to divide. And we'll talk about exactly what this stage is in a minute, but that's why you usually see pictures of chromosomes looking like this. And normally, most of the cell cycle, a chromosome is just 1 strand. So we know that DNA is a polymer of nucleic acids. There are 4 bases in DNA, A, C, T, and G. It's a double helix. Those strands run in anti-parallel or opposite directions. And a given strand can be extremely long. A human cell has about 2 meters worth of DNA in it. And it's very important to wrap and pack that big, long, skinny strand carefully in order to divide those chromosomes between the daughter cells when they're being produced. Before cell division, each chromosome needs to be copied or replicated, and those 2 copies stay attached until the moment of division during mitosis, and that's what you see here is those 2 strands attached to each other. The place at which they're attached is called the centromere, and that's a specific DNA sequence that allows for proteins to bind to the DNA and keep those 2 sister chromatids attached to each other. There's also a structure that isn't labeled in the book, but I think is important to note, and those are telomeres. Telomeres are on the ends of each chromosome or chromatid. They're, again, just a specific sequence of DNA. You can't see them by looking at it. But their job is to keep the DNA strands tightly wrapped together at the ends. They're sort of like those little plastic things on the ends of your shoelaces that keep your shoelaces from fraying. So they're important in protecting the ends of chromosomes. So this is the typical image of the cell cycle. And the cell cycle begins just after this cell has been produced by previous cell division. And the first phase of the cell cycle is referred to as G1. Sometimes you'll see it referred to as gap 1, which I think is misleading because it's not a gap. This is an important phase for the cell, particularly for the cell to recover from the process of cell division. It's going to grow during this period, and it's going to perform whatever functions it performs in the body if it's in a multicellular organism. At the end of G1, there's an important decision which isn't noted on the figure that's in the book. And this is when the cell needs to make a decision about whether to continue through the cell cycle and divide, or whether to pause and just continue to do whatever its function is as a cell. So we'll talk more in the next video about why cells make each choice, but essentially we're going to go through the cycle for now, and then we'll see why a cell might choose to go one way or the other later. So if cells do not need to divide for whatever reason, they're going to go into this G0 phase. Most cells spend most of their time in G0. This is, again, when the cell is doing its job. It's not preparing to make new cells, but it is just simply performing its function. If that cell needs to divide, it will go into S phase, and S stands for DNA synthesis. This is when the DNA is copied. And there are other things going on in the cell at this point, but DNA synthesis actually takes an enormous amount of energy, so a lot of the other cellular functions do slow down during this period. Once this DNA has been replicated, the cell is committed to dividing, or it will eventually die. It's not good for mammalian cells to have more than 2 copies of their DNA, and so it will need to divide eventually. The next phase is called G2. This also involves more growth. Usually organelles are replicated and divide during this period in preparation for cell division. Mitochondria and chloroplasts actually kind of divide on their own schedule, and you might think about why that might be. But they need to finish dividing by the end of this phase. Additionally, in this phase, the centrosomes form, and those will be important for the process of mitosis. So collectively, G1, S, and G2, and actually G0 as well, are known as interphase. And interphase is kind of also a misnomer. It sort of implies that nothing else is going on, but this is actually the majority of the time of most cells. And these last phases here are mitosis and cytokinesis, and we're going to look at those in more detail. Those involve the division of the chromosomes and the cell materials.